Hello everyone, welcome to the another video tutorial. So in this video lecture, we will discuss about chemotaxis. So let's start with the introductory part. So the word chemotaxis is made up of two words, where the word chemo indicates a chemical substance which is a form of matter which can be either solid, liquid or gas, having several constant chemical composition and some characteristic features. The word taxis means the movement of any organism in response to a stimulus like light or presence of food or many other stimulus, and that's why the chemotaxis is considered or defined as the movement of any organism in response to the chemical stimulus. So the chemotaxis is a movement or response of any organism to the particular chemicals which are secreted by several organisms or several other bodies. Now this is the phenomenon in which any bodily cells which can be prokaryotic or eukaryotic, they can be bacteria, any other some singular or multicellular organisms, and in this phenomenon these organisms move according to a certain chemical. Now let's understand about the history of chemotaxis research and about the discovery of chemotaxis. So basically, the migration of cells was detected from the early days from the development of microscopy. We know that Anton von Leeuwenhoek discovered the microscope and he also discovered about the bacterial cells. So the description about chemotaxis was first made by T.W. Engelmann and W.F. Pfeiffer independently in 1881 and in 1884 respectively. Later on, in 1906, a scientist named as H.S. Jennings described the chemotaxis in ciliates, and the pioneer work of J. Alden modernized the Pfeiffer's capillary assay to describe about the phenomenon of chemotaxis, and he also represented a significant turning point to understand the whole process of chemotaxis in the intracellular signal transduction in bacteria. So this is the history and discovery about the chemotaxis and chemotactic research by different scientists. Now the chemotaxis can be of two types. So basically it are of two types, including positive chemotaxis and the another one which is named as negative chemotaxis. So basically in the condition of positive chemotaxis, the organism move towards the chemical stimulus, and that's why it is called as chemotaxis, and this is called as chemoattractant. And in the negative chemotaxis, these organisms repel the stimulus and they move opposite to the stimulus, and these stimulus are called as chemorepellent. And these chemoattractant and chemorepellent are organic or inorganic substances which possess several chemotaxis inducer effect in motile cells. So these are the types of chemotactic movement or chemotaxis. Now at first, let's understand about the bacterial chemotaxis. So as we have discussed in our earlier slide, that chemotaxis can occur in all the organisms, in bacterial cells as well as in eukaryotic cells. So let's understand about the mechanism of bacterial chemotaxis, because the mechanism of bacterial and eukaryotic chemotaxis is different from each other. So we know that bacteria are type of prokaryotic organisms which do not have a definite cell structure and they are unicellular in nature. So there are several bacteria which have a particular organelle named as flagella and cilia, and these organelles help in the movement of these bacteria. So some bacteria like E. coli have several flagella in a particular cell, which means they have more than one flagella in a single cell. So these flagella can rotate in two ways, like counterclock or anti-clockwise, and the another movement is clockwise. So in the anti-clockwise rotation, they align the flagella into a single rotating bundle, and that's why they cause swimming of the cell in a straight line. But in the condition of clockwise rotation, the flagella break, bundle apart, and each flagellum points to the different directions. They point to the different directions in a round way, and that's why in that condition they cannot swim, and they cause the bacterium to tumble in a particular place. So what happens if a bacteria get any type of chemotactic presence, or in the presence of any chemical, bacteria will chemotax. So in that condition, several bacteria which have single flagellum adopted a different method for chemotaxis, but these bacteria which have more than one flagella in the structure, they move to the straight line, or they swim with the anti-clockwise rotation if they have chemo-attractant chemical. But in that condition, when they have chemo-repellent, they adopted the clockwise rotation and cause the bacterium cell to tumble in the particular place. So this is the mechanism for the bacterial chemotaxis. So now let's understand about the eukaryotic chemotaxis. So basically the mechanism of chemotaxis in eukaryotic cells is quite different from that of the bacterial cells like E. coli and other bacterial cells, but the sensing of chemical gradients is now also a crucial step in the condition or in the process of eukaryotic chemotaxis as it was in the prokaryotic chemotaxis. But due to their small size and other conditions, E. coli cannot directly detect the concentration gradient of the chemical stimulus. But in the eukaryotic organisms, they are slightly larger and much larger than prokaryotes, and they also have a well-defined cellular structure. So basically the eukaryotic cells have several receptors which are embedded throughout the cell membrane. We know that the cell membrane is the outermost covering of the eukaryotic and prokaryotic cell. So the eukaryotic chemotaxis involve detecting the concentration gradient by comparing the asymmetric activation of these receptors in the cell wall or in cell membrane. So with the help of activation of these receptors, it results in migration towards the chemoattractants if they are chemoattractants, and if the chemical stimulus are chemorepellents, they move away from these chemorepellents. So it has also been shown that both the prokaryotic and eukaryotic organisms are capable for the chemotactic memory, which means that they have the chemotactic memory and they have the ability to recognize the particular chemotactic stimulus. So in the prokaryotes, this mechanism involves the methylation of different types of receptors, but in the condition of eukaryotes, they adopt a different method. So this is the process of eukaryotic chemotaxis, where the chemotaxis is slightly different from the prokaryotes because of their well-defined structures and because of their receptors which activated, and the receptor activation results in migration towards the chemical attractants, and in the case of chemical repellents, they move away from these chemorepellents. So now let's understand about the significance of chemotaxis and about chemotactic movement. So the chemotactic movement and chemotaxis is very important in prokaryotic as well as in eukaryotic organisms, and the significance of this phenomenon is very significant in the humans as well as in other animals and plants also. So in humans, the immune system of humans, which include white blood cells, uses some cells like neutrophils and macrophages. These neutrophils and macrophages are part of these WBC, and they have the ability to identify and to detect the infectious agents, which are also called as foreign particles, and they have the ability to catch them or to engulf them or to destroy them. So they use the chemotactic movement or the chemotaxis to identify the infectious agents with the help of chemical stimulus produced by these infectious agents, and that's why they use the chemotactic movement for their mechanism. Now in the case of microorganisms including bacterial cells, these bacteria use the chemotaxis to detect toxins. So basically, in that case, they detect the chemical stimulus produced by those toxins and they move away from these toxins, which is called as chemorepellent movement. Beside this, these microorganisms or bacterial cells also used the chemotactic movement to find the food source. So basically they find the food source with the help of chemotaxis and they move towards the food source, which is called as chemoattractant, and this movement is due to the chemotaxis. Now the chemotaxis have also some clinical significances in the several diseases. So basically there are several endogenous chemotactic ability of the microorganisms by which the pharmaceutical agents can decrease or inhibit the ratio of the infections. Or apart from these infections, there are several other diseases. They can be genetic or some other dysfunctionings where the impaired chemotaxis is the primarily etiological factor. The most common example is the Celiac-Higgs syndrome. So the Celiac-Higgs syndrome, which is commonly also called as CHS, is the rare autosomal recessive disorder which arises due to the mutation in the lysosomal trafficking regulator protein. So basically it is a type of lysosomal dysfunctioning occur in humans and it led to the decrease in the mechanism of phagocytosis, which can lead the infections in the particular organism. So in this disease the chemotaxis is also used. So there are different other clinical significances like in different diseases like in inflammations and some other like arteriosclerosis and arthritis. It is noted that in the patients which are suffering from those diseases, the chemotaxis is increased. And there are several other diseases like AIDS, Celiac-Higgs syndrome, multiple sclerosis and many other diseases where the patients have decreased rate of chemotaxis which are suffered from those diseases. So with the help of diagnosing or measuring the chemotaxis, we can diagnose the disease in the particular patient. So this is the clinical significance in animals of the chemotaxis. Beside animals, there are several plants which have also some significances of chemotaxis. So the plants use the chemotactic movement in the process of pollen grain movement. So with the help of chemotaxis, the stigma is moved towards the pollen grains to complete the fertilization process. So basically these are the clinical significances of the chemotaxis and chemotactic movement. So in this video we have discussed about the chemotaxis, types of chemotaxis, mechanism of chemotaxis in eukaryotic as well as in prokaryotic cells and different significances of this phenomenon. Thank you.